Today's topic is salient achievements in crop biotechnology. So basically we are going to define here the application part of biotechnology as far as crop improvement is concerned. So the field that discusses the achievements of biotechnology in crop improvement, that is called agricultural biotechnology. It is the collection of scientific techniques used to improve plants, animals and microorganisms to enhance their value. So this is the basic one. Whatever plants, animals or microorganisms are related to agriculture, okay, because we have a use of cattle in that. Many microorganisms, they are beneficial, some are harmful also. So all these, whatever is related to the agricultural land, when we study various techniques to improve the overall agriculture, that study comes under agricultural biotechnology. It increases agricultural productivity, then it enhances breeders' ability to make improvements in crops and livestock. Whatever breeding programs are done so that the crops or the livestock, means cattle, buffaloes, etc., they could be improved. That is under agricultural biotechnology. Enables the improvements that are not possible with traditional crossing. Sometimes the species which are related, they does not produce a desirable hybrid if we go via the conventional breeding methods. But this is a technique which just overcomes all the complications and it can help you generate hybrids from different species or genera level. So it makes that improved variety formation quite easy. Now, this is a history about agriculture. Okay, so the first is the agriculture began around 12,000 to 4,000 BC. First use of grafting, where the plants, means cayenne and the calf part, they were mixed. So that was done in 100 BC. That is very early. This technique of grafting, it started where the characters of two plants, they can be mixed. The sexual reproduction was discovered in 1691, and the hybrid maize production, that was studied in 1900 year. So here basically the maize variety that was produced via the conventional breeding method, hybridization, the fertile variety that was released. Then first protoplast fusion, that happened in 1909. So scientists, they developed the techniques. We have already studied how the protoplasts are isolated and they are fused. Then there came the era of mutation breeding. So the genes which are whose expression is to be changed or who are to be silenced, all those phenomena, they were under the mutation breeding, and the use of different rays and chemicals that started for mutations. The first one was the use of X-ray, and that started in 1927. Then there came the technique of plant regeneration, that comes under PTC, where the isolated cells, they are regenerated into complete plants, and this technique came in 1967. Then was the onset of genetic engineering, that is the rDNA technology, and first rDNA molecule was formed in 1973. Then first genetically modified plant was released in 83, and first genetically modified food crop was approved in 1994. So that is a bit about the history. Okay, for your question in which you are to explain the achievements, you can just briefly mention if you have enough time, otherwise you can skip this section. So now coming to crop biotechnology. Crop biotechnology itself comes under, this is a smaller aspect, smaller level of the agricultural biotechnology, because there we are concerned about animals, microbes also. But when we are talking about only the crop part, that is called crop biotechnology. Here the desired traits are exported from a particular species of crop to an entirely different species. So we are going to use this technique used to make rDNA of the two unrelated genera or species, and the gene of interest, that is responsible for a desired trait that is mixed to our crop. These transgene crops, they possess desirable characters in terms of flavor, color of flowers, because let's suppose that crop is important in floriculture. So that can also be used to change the color of flowers, growth rate, size of the harvested products, and resistance to disease and pests. So for all these things, the biotechnology is being utilized for crop improvement. Now, what are various techniques for modification? First is traditional breeding. This is the one in which crossing was done. So it started centuries ago, and that was a conventional method to improve crop quality and quantity. So in traditional practices, what we used to do, pollen from one plant were taken and they were placed on the female part of another plant, another species whose characters you want to be mixed in a hybrid. Then the hybrids which were having the desired characters, they are selected and then they are raised and maintained for many generations. And finally, that variety is released into the market. So that was traditional breeding. It took many years. Second comes mutagenesis. So here we are inducing mutations in DNA of an organism. In nature, it can happen randomly, right? We are under the continuous exposure of the UV rays, many chemicals. So they are randomly generating mutations in the DNA of different organisms. But that technique that we have learned from nature, that we can also induce the mutations which are beneficial to plants. So scientists, they have used different agents like ethyl methane sulfonate or different radioactive radiations to induce mutations in DNA to either silence the genes having the undesirable properties in the product or to overexpress few genes. Okay, so gene manipulation has been done basically. Then comes genome editing. Genome editing, this is a use of an enzyme system to modify the DNA directly within the cell. So here we can directly modify the DNA. For example, if there is a wild cultivar or if there is some bacteria or any other organism which is having certain genes for resistance, disease resistance of a type, and I want to introduce that into my crop so that my crop also become resistant to that pest, it can be done. That will be genome editing. Similarly, if I want to use herbicides in the field so as to kill the weeds, but I don't want their effect on my crop of interest, then herbicide resistance genes, which are usually present in the wild cultivars, they can be taken from there and introduced into the my crop of interest so that it becomes herbicide resistant. So that all part where the genome is actually changed, that is called genome editing. We have raised canola plants for this purpose, which are herbicide resistant. Next comes polyploidy. So this technique can also be used. We can modify the number of chromosomes. Whole set can be doubled, tripled, multiplied many folds, and that may ultimately result in the increased size of the product. So polyploidy can be induced to modify the number of chromosomes in a crop. Naturally or with the use of chemicals, we can do that. For example, we use colchicine. Because of that, the chromosome sets, they increase. So number is changed, that result in the size modification of that crop product. It may be a fruit, maybe a grain. Similarly, the seedless fruits, they have also been released in the market, that is via polyploidy. For example, seedless watermelons. Next technique is protoplast fusion. Means we are fusing protoplast. So we are joining the cells of two different species so that their traits, they get mixed. Now in plants, this technique has specifically been used to transfer male sterility. So the male sterility, that helps plant breeders to make hybrid crops. Let's suppose I am having a crop, let's suppose it's plant A, and this is having, the flowers are having the male segments as well as there is a female segment present, that is the ovary, and I want to cross it, I want to use it for the breeding method, so I'll draw pollen of some other species on this. Now these may create problem, right? Because they may also fall on their own stigma, so it will result in a problem. So what if I make this species male sterile? So if it is male sterile, stamens may be present but they will be aborted. No pollen grain formation would be there. So I can easily use this technique. I can easily mix the pollen grains of some other species over the stigma of this species that I desire to modify. So the technique overall procedure becomes quite easy. So that is why the conventional breeders, they are using this technique via protoplast fusion. They are transferring the character of male sterility from one strain to another, so that this extra part of the uh, removal of the anthers, which is called emasculation, that could be avoided. Then comes RNA interference. This is quite a recent technique. RNA interference or RNAi technique, we call it. I is always written as small letter. This is a process in which cells RNA to protein mechanism is turned down. We all know that DNA, it transcribes into RNA, right? RNA translated into proteins. So if I knock off a particular pathway of conversion or translation of the proteins from RNA, if I switch off that gene, so the product will not be formed. When product is, this may be harmful trait for the plant. So a particular gene can be identified which is to be silenced, and through this RNAi technology it can be easily done. Then comes transgenics. Transgenics means when I am mixing the genomes, I am taking a piece of DNA from one organism and transferring it to the another, that will become a transgenic. So why this is done? This is done to introduce new genes into the original organism so that new characters may be introduced or desired traits may be introduced into that. Now coming to the applications part. Let's suppose you get a full mark, 7-mark question on the applications or role of biotechnology in crop improvement. Then you will give a brief introduction about the techniques and then you will directly come to applications. But let's suppose you get just a 3 or 4-mark question on this. You will directly start from here and the points should further be reduced. The content should be reduced. Now what are the applications? So here we will have the examples from the history where the successful improvement of the crops has been done by using biotechnological aspects. First is insect resistance. It's a very common, very highlighted application of biotechnology where the resistance to insects, pests, that has been introduced in the crop, and the genes for that has been obtained from a bacteria which is called Bacillus thuringiensis. By natural means, it has the properties in it, its genes, they produce the proteins which can kill the, it is typically used for cotton, so for bollworm. Okay, so it can produce the proteins which induce gut toxicity in those insects. So we have taken the gene from Bacillus thuringiensis. That's why abbreviated as BT. So that BT gene is introduced into our crop of interest. The first one was cotton. Later on the work was done on corn, tomatoes, tobaccos, number of crops they have been introduced with BT gene. So ultimately, so that this crop now becomes resistant to bollworm. Now this is a typical example. It is the BT cotton and this is a normal cotton. You can see the yield has increased. Why? Because this was infected by bollworm. But this could not be infected. The bollworm, when starts feeding, see this is a bollworm. When it starts feeding on the ball, on the fruit of this plant, then because of the chemicals present in the genes now which are going to release BT toxin, it will cause toxicity, gut toxicity to these bollworms. These are called bollworms, and finally they will die, so that there is no damage to the flower or the reproductive parts inside, and ultimately the produce increases. Okay, so this is how it is done. From Bacillus thuringiensis, the gene for BT toxin is taken out. It is inserted into the cotton cell. So there is a formation of rDNA so that the toxin gene starts expressing in the cotton cells. And when the plant or the is attacked by the bollworm, this toxin which is already there in BT cotton, this will cause gut toxicity in the bollworms and they will die. Okay. This has been quite successful in the market. Many farmers, they are now sowing BT cotton. The produce has also increased, but some, because The use had been at a very rapid pace, because of which now some other problems are getting created. So this was mainly for cotton bollworm, the black worm, but now there is another resistant variety that is resistant to Bt toxin. It has developed, which is a pink bollworm. So this problem is arising in certain areas now, where the Bt genes are not going to kill these worms. Okay, so this is happening. Now, second comes herbicide tolerance. So we are actually introducing the tolerant genes into the crop of interest, so that when we spray herbicides over the crop of interest doesn't get affected. So weeds, they have proven to be an issue for the farmers for thousands of years, because they are going to compete for nutrients, water, sunlight. So that's why they put forth a strong competition for crops. And biotechnology, it has offered a solution for that. What is that? We can use chemical herbicides to kill the weeds, and those herbicides, when they fall on the crop of interest, because they contain resistant genes, so they will not be affected and they will flourish properly. This has been seen in case of many grain crops, tobacco, soybean, etc. This is an example of the bean plant. You can see this is a resistant crop, and this is a crop which was sensitive to herbicide, so it got affected. Third is disease resistance. So this can also be introduced. The genes can be identified from the wild cultivars, and they can be inserted in the crop of interest. And this is done for many pests, for many insects which attack the crops at a large scale and totally devastate the land. So it has been very helpful for that, as well as for the viruses also, because viral diseases, they spread at a very rapid pace in the crop. So it has been done for that purpose in cassava, maize and sweet potato. Next is temperature tolerance, or any kind of the abiotic stress tolerance you can write. So you can replace this with abiotic stress tolerance as well. So the genes which are related to any kind of the tolerance, maybe salt resistance, temperature resistance, drought resistance, they can be identified and introduced in the particular required areas where it is required. For example, plants which are grown under very cold and hot environments, the temperature resistance gene can be inserted in that. It has been done for papaya. Similarly, if the plants which are growing in the desert areas, which have to face the drought conditions, there the drought resistance genes can be introduced, and so on. Quality traits. Quality traits now include number of characters, means we can have better color, better vitamin content, better size, better digestible carbohydrate component in the crop. So number of facets, they come under quality traits. These increase the nutritional or the dietary value, improved food processing or storage for a particular crop, that can also be done. Elimination of toxins from that, if by nature they contain certain toxins, they can be removed, like tannins, etc. They are toxic. Then allergens in a particular crop, they can be removed via the biotechnology. So here we have raised transgenic crops like soybean for high protein content, potatoes for more nutrition, beans with more essential amino acids. Okay, so this is a Bt potato. You can see here, this has got more nourishment now. It has abundant anthocyanin content, and this is a normal potato. Now few more examples, like flavor saver variety of tomato has been raised so that its decay is delayed. So the shelf life of the plant product increases, shelf life of tomatoes. So that's why they can be easily transported, and their decay will be delayed, so they can have nutrients for more abundant nutrients for longer duration. Second is transgenic peppers and melons. They have been raised in order to have better flavor. Golden rice varieties have been released, where you can see this is a golden rice, this is a normal rice. What is special about this rice? This has got a gene for vitamin A production. So all those where there is areas where there is VAD, vitamin A deficiency issues, there these varieties are being raised. Then bio cassava, that has been raised because originally cassava contains the toxins, that is cyanogen glucosides, but this variety bio cassava, this has been raised so as to reduce the level of cyanogen glucosides and enhance protein and other nutrients in that. So toxin reduction can also be a part of quality improvement. Now next aspect is tissue culture. We have studied number of applications separately. The few are enlisted here. So tissue culture is a mean where we can regenerate plants, we can have disease-free plants, and there can be mass production. This has been practiced for in citrus, pineapple, mangoes, many crops. Okay. Then from tissue culture, certain variations they appear in the clones. They are called soma clonal variations. If the variations are desirable, if those variations are beneficial, then the cells they can be harvested and they can be cultured and regenerated. So it gives good characters or variations in the clones. Then embryo rescue. Sometimes the hybrids when raised, their embryos they abort at very early stage. They are unable to grow within the ovaries of the mother plant. So in that case, those hybrid embryos can be excised from the ovary and they can be cultured separately on the artificial medium so that the chances of their survival increase many-fold. This is an example where from the seed the embryo has been taken, and this embryo, see how on the artificial medium this embryo started propagating and ultimately a complete plant is raised. Then intergeneric and interspecific hybrids, they can also be raised via the technique called protoplast fusion. Another aspect is environmental benefit. Now, what is the fun of using biotechnology if it is not giving environment friendly output? One, we have considered the herbicide resistance, but that led farmers to use abundant herbicides. But this technique, moving towards the path of environmentally eco-friendly, eco-safe techniques, this has now started producing the genetically engineered crops which have less reliability on the pesticides or herbicides, okay, which are having more stronger use of the resources as compared to the weeds, or which are pest resistant. So even if the pest attacks, for example, the BT crops, so now it is moving towards the environmentally friendly approach where there is reduced reliability of the crops on pesticides. When there is less use of pesticides, so though they will not leach out and mix in the groundwater, because of that the underground resources of the water they will remain safe to use, and so that's why this is offering an environment friendly approach. Next is biofuels. Biofuels, this is again a very important topic of discussion these days, because our the non-renewable resources, petroleum, etc., they and coal reserves, they are depleting day by day. So we are looking for the alternates, and for that the crops are being sought after in which the biofuels could be raised. So if for that algae is being harvested, especially microalgae. After genetic modification, if it can yield more of the fatty substances which can be used for biofuel production. Then maize, there is a maize which is trademarked as Enogen, that has been genetically modified so as to convert starch to sugar for finally ethanol production. So the engineering of the crops is being done so as to raise biofuels. Next is bioremediation. This is again an environment friendly approach of biotechnology where we are engineering the species. There may be a weed species, maybe a plant species. We are engineering the species so as to reduce contaminants. Bioremediation means they will sequester those harmful chemicals within their body. They may reduce the effect of those harmful chemicals. It can be good for the contaminated soils where a lot of mercury and arsenic, lead poisoning is there. So that can be taken up by the genetically modified plants. And similarly, for the water bodies also, if there are a lot of contaminants, for example, the areas where the factory discharges, they are released into the water bodies, there also we can use these cells, especially the algal cells, which are genetically engineered, which will convert toxic products into the non-toxic form. See, you can see here, these are the bacteria which are making coagulates of the toxic substances. So that is beneficial for our water resources especially. Last is drugs. The drugs, they have also been somehow the genes which are used for treatment of particular diseases. They like we have engineered tobacco plants. In the tobacco plants, the genes have been introduced which will produce antibodies for the disease Gaucher's. Gaucher's disease is when the fatty substance in the body organs start getting deposited, especially the spleen and liver is affected. This is a hereditary disease. So it is not the one which is related to the errors in metabolism. This is actually a hereditary disease where the fat or you can say recycling of the fatty substances is not there. So the fat it start getting deposited in the body organs, especially in the liver and spleen. Now it can be treated with the antibodies, and those antibodies, the genes responsible for that has been engineered in tobacco plant. So people directly while consuming these tobacco plants, they can have solution for this Gaucher's disease. They need not depend upon the allopathic preparations on the drugs. So this is the plant itself, they will serve as drugs. So this is a very, very recent achievement which is being, and now other crops, they are under the trials in which such techniques they could be utilized. So that's all about the application part. Now comes what are the issues related to that, because people, they are very biased. They have different opinions as far as the mixing of the genomes is concerned. So the genetic engineering, direct one. Direct means when I am raising transgenics. When I am having a gene of interest inserted into a vector and then into a microorganism, people don't have any problem with that. But yes, when I am mixing the genomes of two different kingdoms, for example, from bacteria to the plants, from bacteria to the animals. So when I'm mixing that, that is the direct genetic engineering, and it is controversial ever since it started. Now, most but not all of the controversies are over the GM foods rather than crop per se, you can say. So, mostly they are related to the foods. They are not related to other articles where GMs have been raised. It is special concerns are related to foods. Now, what are the major objections? The one is the environmental impacts of these using mixing the genomes, and if by chance the gene get lost to some other plant, some other animal, if it gets transferred to that, so what will happen? So we are not sure about the stability of that new genetically engineered genome. Then food safety issue. There are concerns on the religious grounds that how can you mix a gene from a bacteria to the human beings or to any other organism. So there are religious grounds also. So other issues, these are the primary issues. There are secondary issues like labeling. Few nations, they are fighting over this concept whether the GM and non-GM crops should be put under a label in the market. That okay, there should be personal choice. There should be a label on that crop whether it is GM or non-GM. I can go for a GM or non-GM, but it should be specified. It should be labeled. But few nations, they say no, they should be considered equivalent. So there should not be labeling or demarcation of GM and non-GM crops. Then behavior of government regulators. So that is another concern, whether you want to earn the revenue, then a particular company is given rights to raise the GM crops. Then it becomes issue for other persons, other private industries to get indulged in that. So that is a government regulator. So these are secondary issues. That the major concern is about the stability of the genome. So a significant environmental concern is about these crops is that there may be possible crossbreeding with the related crops. Let's suppose I am inserting the Bt gene in the cotton and somehow it escaped and it entered into some other crop. Let's suppose radish, turnip, any other crop which is growing during that season or in the vicinity. What will happen if they undergo natural crossbreeding? So that gene was not intended for that crop, but it entered the other one. So there are issues related to that. Then there is a scientific consensus that currently, whatever information we have from the history, because this is a practice which is around 2-3 decades old. So whatever evidences are there from the history, the scientists, they have validated that GM crops, they pose no greater risk to human health. Right now, till date, we don't have any such information that a gene has outcrossed some other non-target crop. No, we don't have any information about that. But each GM food, that need to be tested first. okay, before getting released into the market, before its introduction into the market, that should be properly tested. This is really important. It is not that, okay, for the financial gains the variety is tested for a very short duration and immediately released into the market. No, it should not happen. It should be properly tested on wild organisms and then only released as a GM food in the market. So that is the major suggestion by the scientific community. So that's all about biotechnology, and you can complete this topic. If you just get a question on pros and cons of this crop biotechnology, then you can mention the issues that we have discussed in the last slide. That pros, what are the pros? They are actually the application part, but you need not discuss that in detail. You can simply write the title with example, just 1-2 lines about each, and then you will come to the cons. Cons is when whatever issues we have related to the environment, related to religion, labeling part, okay, that you can point wise mention 3-4 issues that are there. So that will serve the purpose about the pros and cons of biotechnology in crops. So that's all for this topic.